Hello, students. Welcome to our discussion on emotion, stress, and trauma. This is using Chapter 10 from our primary book and then Chapter 12 from the OpenStax neurobiology book. So lots of material in both of those, and we'll focus on a couple key things and go beyond some of the readings as well in some of these cases. To begin with, I wanted to share this comic. Depending on where I will put this lecture, this may be close to your final exam coming up. You may be feeling stressed, and so learning about stress while you're feeling stressed can increase your stress. One of the in-class activities we'll do is a mindfulness exercise, but do things that help you decrease that stress. Take breaks from work, things like that. With that, maybe you can remember the things we discussed on how to learn and remember things. Um, Taking breaks every about 30 to 45 minutes, and even switching to another class in between can be a very good way to remember things, but it can also help you with your stress as well, especially if what you're doing in between those times is something that you're enjoying. So, to begin with, before we get into stress, let's talk about a lot of emotions. So, here in this picture, there are tons of emotions, and we'll talk about where those come from. But first, a little bit of definitions. So, affect essentially means feeling. And with English, sometimes you'll see affect and effect used incorrectly or interchangeably. But affect, when we're talking about psychology, affect is a feeling. So, affective neuroscience is neuroscience of feelings. We're studying feelings. We're studying all these feelings on this chart. So, in humans, most of the studies that we do are using EEGs, are using fMRIs. So, the CAPS, for example, and fMRIs. And sometimes we're doing things like injecting people with adrenaline or trying to increase positive emotion. So, maybe watching a video clip. Giving someone a gift is another way that we try to increase some positive emotion. And what you'll see is that in your book reading, you've probably already seen, hopefully, the material on how we study this in rats and mice. We do a lot more kind of terrible things and invasive things to rats and mice to make them feel things. Oftentimes, these are fear and stress responses. As we'll discuss in a minute, a lot of the fear and stress responses, one, is a lot easier to see the behavior in rats and mice. So it's very much more obvious that they're feeling stressed or they're feeling fearful, versus it's really hard to tell if a rat is feeling happy. So that is why some of these studies of emotion really focus on these kind of negative feelings because they're so central to our survival. They work very quickly. It's a little easier methodologically to investigate emotions that are negative, like trying to get you away from a situation. But this is how we started to learn about a lot of different emotions. So in humans, again, we're looking at what is active in the brain. And again, there are limitations to what the EEG or the fMRI can show us, what kind of situations you can be in when you're doing that kind of sensor. And then also there's limits to how invasive we can be with humans, for example. If you looked at some of the boxes, I think this was in the OpenStax books, there's examples of people who, as a form of treatment, do have electrodes implanted into their brain, and it's activating specific areas, and we see that that has related to more positive or the lack of negative emotions, things like that. But we don't do that in studies of humans, but we do that in studies of rats. We can implant things into their brains. We can make them fear that they're going to be drowned. We can isolate them socially. We can strap them up by their tail to the roof of a cage. There's a lot of things that we do to induce emotions. Mostly for humans, though, we're talking about things like watch a fun video or something like that. And we'll do a little bit of that today, the watching fun videos to try to, in the in-class portion activities, we'll watch fun videos to try and induce positive emotions and maybe some more negative emotions too. So with this, emotions and emotional responses are organized around any given organisms' survival and reproductive needs. So essentially, anything that has a direct bearing on the organism, so that rat or that humans, so the human's goals, needs, values, general well-being could elicit an emotion. So anything that has a bearing on those. So it can be elicited from a wide variety of things. And emotions are in multiple brain areas, so multiple circuits. And this is something that we've seen kind of consistently from the first week is we've saw the basic areas of the brain, but any given function that we're talking about is more than just one specific area. It's a bunch of areas and networks working together. So a lot of the study of these emotions is which which areas interact in series. Like, what is the circuit? What are all the areas that are working together? And is there one that starts first and then this one is last? That kind of thing. Oftentimes, the prefrontal cortex is one of the last involved in emotions. So um, most of the emotions basically start in the middle of the brain. So thalamus, hypothalamus, amygdala, this kind of area is kind of where a lot of these emotions begin, their first step. So how many basic emotions are there? Well, it really depends on which researchers you ask. So these are the ones that our main book primarily talks about: desire, liking, fear, rage, love, and grief. And the main reason the book takes, and a lot of researchers take the perspective that these are the basic emotions, is that these are things that we can detect essentially immediately after a child is born. Immediately after a child is born, we can see evidence of desire, we can see evidence of liking, we can see evidence of fear, we can see evidence of rage, we can see evidence of love, and we can see evidence of grief, both in like the behavior of a newborn infant, as well as in the neurology, like scanning them with an EEG, like put a cap on them. We can see that there's different brain patterns basically immediately after being born. So it seems like these are core basic emotions. So that being said, these are not the only argument for core basic emotions. So I will jump forward in slides really quickly. So other studies suggest maybe these are the core basic emotions. And the main idea here is that when you ask people cross-culturally to identify the emotions in these faces, everybody around the world can identify these as the same thing. So maybe these are basic core central emotions to humans. So because all humans across the world can identify these emotions. With that being said, cross cultures, there might be different social rules for how do you or when is it appropriate to display happiness, right? So if you're talking about abuse and trauma in a classroom, people laughing at that is probably not culturally appropriate, right? Or is it appropriate to show anger to a boss in a situation, right? Depending on the cultural rules, you might have different ways that these display in social situations, but people cross-culturally can identify and use these emotions and have the same facial expressions. So that's kind of the argument for maybe these are the basic emotions. And even more than that, we'll talk about this in a little more detail. This is a little bit additive. So talking about these as basic emotions. So these are things that people can identify. We also see some different body reactions. So we'll talk about this in more detail in a minute, but here's an example of some different body areas, temperature maps, of what areas of the body are active for these different emotions. So those body systems, we can see kind of consistent patterns. Maybe that is what all emotions are built off of. The main idea is we're still getting a lot more clear with those emotions. We will go from the argument of our main primary textbook that these are the basic emotions. These are things that we do see clear evidence neurologically that there are different kind of patterns and pathways for these to be active in, and they can be seen immediately after birth. So that's a pretty cool thing, and a good kind of argument, I think, especially in a neurobiology class, to focus on these as the basic emotions. But we'll talk about the other ones as well. With that, let's start with the emotion of desire. So you could think of desire as reward seeking, wanting to get rewards, seeking for things, wanting to do things. So oftentimes it looks like in, say, later in life, like enthusiasm or interest or curiosity. So if you want to learn about something, if you want to go and find bugs outside, if you want to go swimming, if you want some ice cream, right? This is showing that desire slash reward seeking system. And that is one basic emotion. So curiosity is a good example of this. If you see this ice cream sundae and you're like, oh, I want to eat that, that is activating this desire slash reward seeking system neurologically. So we seek other things too, appetizing foods, sex partners, pleasurable stimuli, and some of the main areas for this are the lateral hypothalamus, the amygdala, nucleus accumbens, and left frontal cortex. So for this specifically, for desire specifically, and wanting to get something, that the frontal cortex is activated, but more on the left frontal cortex. So depending on if I have my camera mirrored, I might be pointing to the wrong side. I'm actually pointing to the left side of my frontal cortex. So hopefully that's not confusing with any mirroring of my video. Yeah, that looks right. So yep, these are the different areas. So we've got lateral hypothalamus, which is not labeled here, but it's up above here. We've got the amygdala, nucleus accumbens right here, and then left frontal cortex. This is the right side of a brain, but the left side. All right, so those are some of the areas that we see specifically in the emotion of desire or reward seeking. And so we see this in infants in desiring to be close to others, wanting food, all sorts of things. So this is one example of a basic emotion that we come out of the womb with, basically. You could think of this, I always use the surprisingly, I think I have no images of Inside Out in my slides, considering this is a chapter on emotions. But you can think of those basic emotions, those emotions in Inside Out 1 that they start off with. That kind of came from this idea of basic emotions. So that is desire. Desire is pretty related to liking, the emotion of liking or pleasure and enjoyment, but it is distinct. So they do have different neural pathways. So again, brain circuits are often shared with other brain circuits, right? So stress and fear sharing some pieces of the brain circuits, but we can identify some different circuitry, some different pathways between that desire piece and this liking piece. So liking is pleasure and enjoyment. So this is more how much reward you are actually feeling when you get something, right? So I actually give you that ice cream. You're eating that ice cream. How much reward are you actually feeling? How much do you like that ice cream? Did I actually give you a flavor that you don't like? Is it actually made from maybe it's a keto ice cream and it's not quite as flavorful as you were expecting or something like that. So how much enjoyment are you actually getting from the thing? So nucleus accumbens, ventral pallidum, and the orbitofrontal cortex are some of the big areas here. Again, some of those are the same areas as we saw in desire, but some of them are new. So we've got nucleus accumbens. Let's find that here close to the front. We also have ventral pallidum. Here it's very close there. And the orbitofrontal cortex is part of the frontal lobe, but it's kind of under it. It's close to your eyes, basically, inside your skull, kind of back up here. So here we get this kind of activity in these specific areas while we are enjoying something. So whether that is drugs, sex, pleasant smells, puppies, this is a system that is active when you are actively enjoying something. So again, some of these are the same, right? So we'll jump back to the slide on desire and see which ones that you can see. So we have still nucleus accumbens is one, right? And then we see ventral pallidum was in both, correct? Or maybe I'm incorrect. Nope, not both. So there are some similarities, right? And that makes sense. If you're desiring something and then you get it, you're having two different basic emotions, but they are very related. Like your experience of them is very related. And the areas in your brain, the brain networks, are still using part of those same networks, but liking and desire are using different neural circuits. So hopefully you can understand those distinctions and those differences as we get to some of these other ones. We'll see some of those similarities that some of those neural circuits are used for multiple emotions, even if they're both using maybe the same core or using other outside or further away neural circuits. So the next emotion we'll talk about is fear. So this is, like I mentioned earlier, one of the most researched, partially because it's a little easier to research. It's a little easier to induce fear because it's so central to survival and adaptability. It's a lot easier to see when a rat is fearful versus if they're really liking this sweet water versus this sweet water. That's a little more difficult to tell, versus fear is a little more easy to induce quickly. So, one thing that we see, especially in rats, is that this motivation to avoid potential harm can lead to different responses. So, you've probably heard like flight, flight, freeze, and so fight, flight, and freeze. So, essentially, one thing that we see with a lot of rodent models is that a little bit of stimulation from this system leads to the freeze response. And then a lot of stimulation leads to the flee response. So if you think of a deer standing in headlights, it's in the freeze. It's getting a slight stimulation to this, all of the sensory organs and the pieces of the brain that are involved, which we'll describe in just a minute, for fear, right? So a little bit of stimulation in the fear area of the brain, versus if it then runs away or jumps away, it's getting more intense stimulation there. Now, this doesn't appear to always be true with humans. We can see also freeze responses with really intense stimulation of humans. So again, the animal models, the human models, they're similar, but they are a little bit distinct. And getting a better sense of that is one reason that we do so many varieties of studies on animals to see how it might be working in humans. But again, doing those kind of invasive studies in humans is not ethical, so we don't do that. All right, so for the fear response, sensory organs are one way that we get this information. So in this case, we see the spider, you see a snake, whatever it might be. So that's a very common sensory organ to incite the fear response. Other one could be audio. So maybe you smell something even. So you smell something burning and you're afraid, oh no, did I leave something on the stove? You hear a sound, oh no, is that a saber-toothed tiger going to eat me? Right, so your sensory organs send that information through the thalamus. So, and that thalamus can go through two different pathways. One of the pathways that it goes through is to the visual cortex, in this case, the visual cortex, like seeing the spider, and then to the prefrontal cortex. So, your prefrontal cortex, your more cognitive abilities, you're like, oh no, that's a spider. And then you can kind of process, is it a dead spider? Is it a live spider? Is it a fake spider that my kids left out? That kind of thing. Or maybe it's just a dust bunny or something like that, and I need to put my contacts in. So that is one path, going from the thalamus to the visual cortex to the prefrontal cortex. It can also, at the same time that it's sending it to the visual cortex and the prefrontal cortex, the thalamus is also sending it to the amygdala, which is involved in a lot of these different emotions, and an area that we haven't really talked about yet. It's called the periaqueductal gray. Yep, I got it right. Periaqueductal gray. So I will show that in this image here. So at the same time that you're getting that message from the thalamus to the visual cortex to the prefrontal cortex, so you're starting to recognize, oh, that's a spider, your brain is already sending it to the amygdala and the periaqueductal gray, which is back here, to start getting to those movement areas of your brain. Do you need to freeze? Do you need to run away? Do you need to fight? That kind of process is starting at the same time that you're getting that conscious recognition of what is going on. So your body is already prepping it. Here, the periaqueductal gray is also connected to other systems like the movement system, to systems in the pituitary gland, and that helps with connecting to hormones in your blood to start charging you up and get your body ready to do something. So with that being said, let's watch this video a little bit more detail on the amygdala. And so fear is one of those that we'll come back to in a little bit when we're talking about stress, but some of these systems are involved in those processes too. But again, before we get into the video of the amygdala, the ones we've talked about so far, desire, liking, fear, these are some of the basic emotions that we see right out of the womb.
Well, the amygdala is a small region of the brain. It's in what's called the temporal lobe of the brain. If we divide the cerebral hemispheres into the four major lobes, occipital, temporal, parietal, and frontal, it's in the temporal lobe, which is under the temporal bone, which is behind your ear. And the amygdala would be located in at a point where a line that goes through your eye and through your ear intersect in your brain. So imagine like someone has shot an arrow through to your eye and another one into your ear. Where those two come together would be roughly where it is, and on both sides of the brain, of course, because most structures, in fact, all structures are bilateral in that sense. So it's sometimes referred to as an almond or an almond-shaped structure, which is a bit of a misnomer because there's one part of it that's almond-shaped, and that caught the attention of early anatomists, and so they named this almond-shaped structure the amygdala. But as the amygdala began to be defined more broadly, it includes a lot of structures other than that little almond-shaped piece. It's most commonly associated with fear, although that's not its only function. It's its most heavily studied function, and the reason for that is that it's much easier to study fear than other emotions. Things that are bad have more weight than things that are good, in a sense. I mean, you can put off eating, drinking, sex, whatever for an indefinite amount of time, but you have to respond to danger immediately. You don't find great novels about happy subjects. It's always about, the bad stuff. And it's sometimes said that there's this negative bias in emotion, which is that most research on emotion is about the negative stuff. But the fact is that the negative stuff turns out to be the most important for survival, in a sense, in terms of immediacy and having to respond to it. So some of the key interconnections of the amygdala, and these connections actually define what it does, in a sense, at least with respect to fear. The amygdala gets sensory information directly from the various sensory systems that process the external world. So the visual system, the auditory system, olfactory, touch, pain, and so forth. All of these kind of come together or converge in the amygdala. And then the amygdala connects on the output side with all the systems involved in emotional reactivity. So when you encounter sudden danger, you might freeze. Your blood pressure and heart rate begin to rise. Stress hormones are released. All these things happen as a result of outputs of the amygdala. So the amygdala you can think of as kind of this, this circle with one input coming in being the input coming in being a sensory flow of sensory information from the external world, and then outputs being connections to systems involved in controlling the responses. So, but we have to expand those inputs, so it's getting not just one sensory input, but all sensory inputs, so each sensory system is coming in. And it doesn't stop there, because in addition to getting information from sensory systems, it also gets information from higher order systems, like the prefrontal cortex and higher order association areas involved in various kinds of integrative activities in the cortex. What it does is a function of what connections it has. So because it has connections with all these sensory systems, it can take in information from the environment of a variety of types and use that information. And so if a sound in the external world occurs right before something painful happens, you associate that sound with the painful event, and that sound will then later trigger a protective defense response. But if the sound occurs just before food, when you're hungry, then the sound will be associated with that kind of a positive or repetitive event. And so what the amygdala is doing is forming associations between random or neutral external stimuli and the kinds of reinforcing events that will stamp in those experiences in a stronger way. So it's creating these what's called Pavlovian associations, stimulus 1 plus stimulus 2, If one of those is in a biologically significant stimulus, then the other one will acquire some kind of biological significance itself, whether it's positive or negative. Now, that information can then be used, and this is sort of what I think you were referring to in terms of adaptive behavior. So, that CS-US relationship, or say sound food relationship, can then be used to guide instrumental behavior, which is goal directed behavior. So, if in the past you've obtained food at a certain location, the stimuli that are proximal to that location serve as conditioned stimuli, and are reinforcing to you because they're close to the actual goal, and they take you towards the goal. Same thing happens with aversive stimuli. It's just that it works in the opposite way.
One thing that video talked about that we haven't really covered in a ton of detail, we've kind of got to it a few times, is those associations, making connections. We do talk about it in the chapter on drugs, but yeah, a lot of this is literally connections in the brain. When one thing happens and then you get a fear response, that can become a trigger for the fear response. So if that sound is happening before food, that might be a good thing. If that sound is happening before you get hurt, then you're going to make that association that something bad is going to happen. I'm going to be threatened by something. So that behaviorism piece is really a neurobiological piece. So it's actually that these networks are learning to be connected. And when that input comes in, well, this network is activated because that's what's happened before in enough times. All right, so another basic emotion is rage. So rage or anger also leads you to either approach or attack something, and it can be evoked from a lot of different things. So physical pain, physical restraint, in an adult seeing something that you see as unjust. But as an infant, we're seeing like physical pain or physical restraint can lead to rage. You might have seen this when trying to say swaddle a baby. They get really upset and really do not want to be restrained. So it is similar in some ways to the fear network. So we still see the amygdala interacting here. We still see that periaqueductal gray. And we see the hypothalamus being involved and the left frontal cortex. So it is some of the same kinds of areas. So this is also connected to some of those circuits that we talked about with appetite, like that desire piece. So appetitive circuits, wanting something. If you don't get an anticipated reward, if you want something and you don't get it, we see that that can also trigger the rage response. So that's why we see some of these same systems. So we've got amygdala, hypothalamus, periaqueductal gray, and left frontal cortex, all involved in this process. We also see specifically some interesting neurotransmitters that are impacted in rage that are similar to things that are involved in pain. So substance P is one that we've not talked about. It's kind of another class of neurotransmitter. It does neurotransmission, neuromodulation, but also testosterone and arginine vasopressin. They are all part of this network. So they are very more active when we're seeing rage or anger approach attack happen versus when we see the desire piece happen or the fear piece happen. So it's not just the brain circuitry areas, it is also some of the neurotransmitters that are unique to rage. And this is really how we've kind of delineated these basic emotions. That yes, the same neural, so this area, the amygdala or the hypothalamus or the periaqueductal gray or the frontal lobe and the left frontal lobe specifically, they're active here, but how are they active? What else are they active with? What neurotransmitters are they using? And this is one thing that we commonly see with rage is that these areas are active with these neurotransmitters specifically. So again, one of the basic emotions. Another one of the basic emotions is the one of love. And this you could also take as care or attachment, like wanting to be with something or someone. So social animals look like we develop attachments. We see it in a lot of mammals. There are other animals in the animal kingdom that are social animals. So it seems like they have similar kinds of systems. And when looking at rats, looking at chimps, they seem to have pretty similar systems as humans for love, care, and attachment. So with this, This goes from anything from warm feelings towards someone, affection. This includes like maternal care, like wanting to nurture someone and help support someone, but also the desire to want to stay close to someone. And you might have heard that word there, desire. It's connected to that system too. So wanting to be close to someone or desire to be close to someone that you care about. We do see a little bit of difference from the nurturing piece to wanting to stay close to people. So those do seem like two unique ways that this care and attachment plays out. So they're a little bit different neurally, but they're using enough of the same system that we kind of bunch them all together as love or care and attachment, even though the nurturing and wanting to nurture is a little different than just the system to want to be close to someone that you care about. So with that being said, some of these areas here, these are more specifically the ones for nurturing. So the love or area for nurturing, we have the dorsal preoptic area at the back of the hypothalamus. So here we go. Dorsal preoptic area is over here. So here's the preoptic area, and dorsal means like on the back, think like dorsal fin of a creature. So dorsal preoptic area and the bed nucleus of the stria terminalis. So that is right over here. I think it might be on the other one below. Nope, it's up here. And then and that area, the bed nucleus, and some of these images put it as BNST. So that bed nucleus of the stria terminalis is a connecting point between amygdala and hypothalamus. So we've got this rear area of the hypothalamus. We've got a connection between the amygdala and the hypothalamus called the bed nucleus of the stria terminalis. And so this is an area for like wanting to nurture. But with that, there are overlap in these areas with things like the wanting to be attached to people. So some of these systems are used for like wanting to be close to people. These are also some of the systems that we see active when people have sexual desire. We also see some similar hormones being used in all of these processes. So oxytocin, argine vasopressin, some endogenous opioids, so those that we create ourselves in our own brain. These are all being used in this kind of love, care, nurturing, attachment, wanting to be close to people system. Again, a basic emotion. All right, so another thing that we see is that this is kind of opposite of what we see in another basic emotion of grief. So this is connected to both loneliness and panic networks. So especially when we're looking at this in infants, we see that infant attachment is also related to separation. So if you're wanting that attachment and is separated as an infant, we see some of these systems activate. And also we see this in adults, some of these same systems with painful emotions of grief, panic, loneliness, if something's pulled away from you, right? That is when we see these systems active. But we see evidence of them very early on in life as well. So the periaqueductal gray, this one right here in the kind of back middle. So we see that it's very close to a pain response area. And I don't remember if I mentioned that in the rage and anger piece. So this substance P is related to pain. Here, grief, we see that there might be some activity in pain areas. Or the dorsomedial thalamus. So that is dorsomedial thalamus. Which one did I have it listed at here? Oh, here we go. So dorsomedial thalamus, the ventral septum, and dorsal preoptic regions. So optic is going to be closer to the eyes over here. And then again, the bed nucleus of the stria terminalis. So back here. So again, one thing that I hope you're seeing in this class as we're getting more into depth of specific brain areas is that while we kind of started off with the first couple chapters of these are basic brain areas like the hypothalamus, the hippocampus, the prefrontal cortex, What we've seen in more and more research is that each of these areas has its own more specific areas. And for any given organism, whether it be a rat or a human, there's probably some overlap. So if it's like these couple millimeters and these couple millimeters in one rat, in another, they might be slightly shifted. But on average, for most organisms, we see a lot of these locations for these specific brain areas do seem to be very genetically tied in the early development of a fetus, an embryo, really. So with that, we see that we can even break down the hippocampus, we can break down the thalamus, we can break down the hypothalamus, we can break down the amygdala to even smaller areas that are kind of responsible for doing their own thing. One thing with that that you can see in this image is we can see this oftentimes in rats. We can probe and activate one very specific area. We can't do that in humans. So we have a lot less complex and specific picture of all of these different specific function areas in humans because we can't do that kind of invasive testing. Things like EEGs, fMRIs, they're getting a broader spectrum. They're not getting as specific view of these things, which is tough when we're trying to figure these things out. But we can kind of guess that we have similar specific systems. And we have seen evidence of similar specific systems in people with injuries or specific tumors or strokes, that specific aspects of whether it be memory or emotion do or do not activate. So even with these big, broader lobes that kind of do a thing, very specific areas of those lobes or parts of the brain do more specific things. And in addition, all of these areas are so central to the very middle of the brain, it's really hard to detect it with like an EEG where you've got the cap that's detecting electrical activity. That kind of specific measurement of an electrical activity that's right, like the person said, with a arrow going through both of these ways, it's a little harder to detect that kind of specifics. So again, grief is kind of the backward system of the love system. So it's using them in another way. It's like that love is being pulled away is what you could think of. And so it's activating some of those same circuits, some of those same networks, but it's using them in a different way. It's still using some of those endogenous opioids. It's using oxytocin. It's also using prolactin, which is often used in liking and loving as well. Panic also seems to be using this system too. Panic is an intense separation triggered by these attachment systems. So these attachment systems say, oh no, that attachment is not here. Panic, I need that. It seems like some of these same areas are doing both the love, care, and attachment and the grief or also panic. One thing that we see interestingly is that testosterone reduces this distress. And so this might be so giving, say, rats more testosterone seems to reduce their panic responses. This might be one reason we see women have more panic attacks than men, because on average, men have more testosterone in their body versus women. And so that is a modulating factor. So it might make it a little less intense in this area. This area is not going to go all the way to a panic attack with a panic response with enough testosterone. So that is some of the basic emotions. We've talked about desire, we've talked about liking, we've talked about fear, we've talked about rage, love, and grief. And so hopefully you can see here that these basic emotions are connected to a lot of the things that we do. And the basic idea that we have is that our emotions are built from these basic emotions, whether it's six, whether it's a little more, like we'll talk about in a second, that our response to the world is built off of these that are kind of genetically encoded to exist, that we have that desire or that wanting for things. We have that liking or we get enjoyment from things. We can have that fear to kind of flee, or freeze in response to danger. We have that system to get angry and attack or approach. We have a system to love and get attached and want to nurture. And we have grief, that if something is removed from us, so an attachment object, we might feel grief for that. So some of these are using some of the very same brain areas. So thalamus, hypothalamus, amygdala, used in a lot of them. I'm going to say it wrong, so I'm going to try to look back on my notes here. Periaqueductal gray is used in quite a few. And hopefully you can see that some of these systems are kind of using it similarly, but with a couple different networks. And then others are very similar, but kind of using it backwards. So with the love and grief example there. And then we see desire liking, pretty similar, a lot of overlap. Fear and rage, a lot of overlap. But from that, the question of how many basic emotions that we have is still a little bit of an open question. Like we said, these are examples of facial expressions that people across the world can identify, and we all seem to use cross-culturally. So that suggests that, well, maybe these facial expressions might be partially genetic too. So maybe our emotional response all the way up to our facial expression is partially evolved and genetic. So in addition to that, we see a commonality that people who are reporting these emotions, so if they're indicating depression, it shows the kind of bodily activity. Is there more blood flowing to that area? Is there more temperature in those areas? What kind of body systems are activated for these different emotions? And we see that the body has some kind of specific ways that it responds to these different emotions. So are these all of the basic emotions? Or are some of these a combination of some of those six that we first talked about and talked about in more detail: the desire, liking, fear, rage, love, and grief? And that is kind of the idea that we're looking at, is which brain areas are active with all of these different emotional responses. Here's a good image. I believe this one actually comes from chapter 13 of OpenStax, if you want to jump into that. It goes into a lot more other emotions there, but that's just too much reading for 1 week of this level of class. And the idea here with all of these emotions is that perhaps some of these other emotions that we experience and we describe and we have names for, maybe what's happening there is that we're kind of putting together multiple emotions, some of those same networks. And since it feels different, we give it a different name. So it might be using the desire and liking system at the same time. Maybe it's using the desire, liking, and grief system at the same time. What would that be called? Right? That's the idea of this kind of image. We say these are maybe some of the basic emotions. So joy, trust, fear, surprise, sadness, disgust, anger, anticipation. So maybe those are the basic emotions. Something that you're feeling very intensely might be something like vigilance. Closer to the central is like the highest intensity of that emotion. So you've got ecstasy, admiration, terror, amazement, grief, loathing, rage, vigilance. And then out here, you've got things that are less intense. So maybe you have submission, awe, disapproval, remorse, contempt, aggressiveness, optimism, and love. So this is still an active investigation of, well, and depending on which researcher you ask, they might have a slightly different perspective of which are the basic emotions. I think there's good arguments for these being the basic emotions since we do see them across all humans. There's good evidence to say these might be all basic emotions, or that these ones that we first focus on in more detail are the basic emotions since we see them right out of the womb, especially since this is a neurobiology class. Focusing on the similarities and differences of these ones, I think, is a really good place to start, which is why we focus so much on them. So with that being said, the next things we'll talk about are a little bit about how do we use these emotions in our bodies. Then we'll get more into stress and trauma. The next thing, though, that we'll look at is theories of emotion. How do you feel them? How does the brain systems interact with the body and hormone systems? Because we've only just barely alluded to that with like the pituitary gland. But emotions are also experienced in your body. That's really what this image is getting at, is that the other body systems are responding too. Your neurological system is not just your central nervous system, even though that's usually what we focus on. Your nervous system is your central nervous system and the peripheral nervous system and the endocrine system. So a lot of these pieces are the peripheral nervous system and the endocrine system being active. So let's see how that works with this video.
Emotions are a tricky subject, both in life and in psychology. Questions like, why do you make me feel this way, are not only confusing on a personal level, but also on a scientific level. First, let's distinguish emotions from feelings and moods. An emotion is an immediate, specific, positive or negative response to internal thoughts or external environmental events. A feeling is the subjective experience of an emotion. It's what we refer to when we say things like, I feel angry, or I feel happy. A mood, meanwhile, is a diffuse, longer-lasting emotional state that doesn't necessarily have an identifiable object or trigger. So if something frustrating happens to you, you might immediately feel the emotional response of anger, and for the next few days you might be in a bad mood. Let's move on to the various components of an emotion. So we know that emotions are immediate and specific responses. These responses have at least three parts: physiological, cognitive, and behavioral. A physiological component of an emotion might be bodily arousal, like a racing heart or tense muscles. The cognitive component of an emotion is the subjective conscious experience of emotion. And the behavioral component of an emotion is the characteristic overt expressions of the emotion. Behaviors that other people can read, like particular facial expressions or postures. Charles Darwin studied this third component of emotions in his 1872 book, The Expression of Emotions in Man and Animals, in which he argued that particular emotional responses tend to accompany the same emotional states in all members of a species. So, according to Darwin's argument, all of the members of a species will display the same behavior when they feel a particular emotion. All cats will display the same behavior when they are happy. And all humans will have the same facial expression and behavior when scared. With this evidence, Darwin suggested that the expression of emotion is genetically determined. This behavior has evolved and has been passed down through generations in our genes. Now let's look at some theories of how emotions work. Let's start with the common sense view. In this view, a stimulus prompts an emotion, which in turn prompts some bodily arousal. So in this case, a frightening spectacle fills our villain with fear, which makes him sweat and makes his heart rate rise. In the mid-1880s, two scholars, William James and Carl Lang, independently came up with similar theories that contradicted this common sense view. James and Lang both suggested that instead of emotions causing bodily arousal causes emotions. So, to return to our previous example, that would mean that the sight of the hero on his threshold makes our villain's heart beat faster, his hands tremble, and his palms sweat, and these physiological changes cause him to feel fear. But this theory is vulnerable to a few criticisms. First, it is possible to have physiological arousal without any accompanied emotion. For instance, your palms might sweat because it's hot, but that sweat won't make you feel nervous, just kind of gross. Second, physiological changes can be too slow to precede the conscious experience of emotion. Our villain might feel scared as soon as he sees our hero, but it might take a few seconds for him to start sweating. Third, the same set of physiological responses are associated with very different emotional experiences. A faster heart rate could mean fear, but it could also mean anger, or surprise, or joy. Finally, there is evidence that physiological feedback is not necessary for emotion. For example, patients with spinal cord injuries who have lost most of their physiological feedback still experience the same emotions as before. However, in the James Lang theory's favor, there is notably some slight evidence of dissipation in their emotional reactions. As an alternative to the James Lang theory, two other theorists, Walter Cannon and Philip Bard, propose that the physiological arousal of emotions and the experience of emotions are entirely separate processes. That is, a stimulus causes the emotion and physiological arousal independently. Neither one causes the other. So the sight of our hero causes our villain both to feel fear and for his palms to sweat and heart to race. But contrary to this theory, there have been many reports of physiological stimuli influencing emotions. So it's unlikely that physiology and emotions are completely disconnected. One suggestion of how arousal and the experience of emotion may be connected but not causal was proposed in the 1960s by Stanley Schachter and Jerome Singer as the Schachter-Singer two-factor theory, which introduces the factor of cognition. The theory suggests that when a stimulus elicits some arousal, this arousal elicits some cognition, which interprets the source of the arousal, and then this interpretation then elicits the experience of emotion. From the stimulus, it takes two different factors, the arousal and the cognition, to elicit the emotion, hence the name Schachter-Singer two-factor theory. For example, say that you feel your face start to flush. According to this theory, you'll automatically use cues from your environment to help explain this flush. You might be mid-workout, or someone might have just confessed their love to you. Although the arousal is the same, the cognitive evaluation of the stimulus is very different across these two scenarios. And so the emotions you would feel would also be very different. Say pride versus shyness, for instance. To return to our previous example, if the sight of our hero makes our villain's heart beat faster, then their cognitive evaluation of the situation, say remembering that the hero is a Secret Service agent here to save the world, or that he is their long-lost love from primary school, will determine their emotional response to the situation. Again, with this theory, it's about the interpretation of the stimuli and the arousal, which leads to the emotion. So let's just quickly review these four theories of emotion to help you remember them. First, the common sense view is that a stimulus will cause an emotion, which causes arousal. Second, the James Lange theory reverses emotion and arousal, suggesting instead that a stimulus causes arousal, which then causes emotion. Third, the Cannon-Bard theory separates out emotion and arousal as independent reactions to the stimulus. And finally, the Schachter-Singer two-factor theory adds another factor to the cause of emotion. After a stimulus elicits some arousal, you cognitively assess the stimulus, and that assessment causes the emotion.
Based on this video, hopefully you're getting a bigger sense of all of the different systems that are involved with these emotions. So with them, the key to remember is that whichever theory you're talking about, whatever we understand now, it is very clear that emotion is not only the brain, but it's also the body's response. So there's evidence of the body making those brain systems work differently or activating them. There's evidence of the brain activating the body systems, but all the systems are working together. So in this class, we focus more on the brain. We focus on things like the hippocampus, the amygdala, the thalamus, the periaqueductal gray, right? But the body systems, the heart beating faster, blood flowing more adrenaline in the hormones across your body, the shutting down or speeding up of different organs, all of this is part of your experience of emotion. And a lot of it is down to your entire nervous system. So central nervous system, peripheral nervous system, endocrine system as well. And depending on what's happening, you can experience fear that might come directly from that. Kind of like what they were saying in the video, the common sense view, seeing a thing, activating those neurons, and telling your body to charge up at the same time as you're recognizing cognitively. But we see variation. But we do have a lot better understanding now of both the neural systems and the body systems that are related to each different type of emotion. The main idea here is it's all connected. Your whole body is a very complex system. The systems that are happening in your hormones and starting your heart beating faster can tell your brain to activate the networks a little bit differently, just like your brain can tell your body to activate hormones in a different way to do different things. So, complex processes. Now we'll talk more specifically about stress. So stress is really a couple of these systems putting together and your experience of that as going beyond what is kind of expected for your body. So to begin with, allostasis is kind of the general expectations, the normal distribution of where your body should be functioning. Obviously, when you're digesting food, the digestive system should be working a little harder, right? Versus when it's not. So both that resting state and that more engaged state are within allostasis. They are within the expected range of activity. Your body has temperature fluctuations, right? So that kind of average, or within that normal distribution piece around that average, is the kind of expectation. Blood sugar has a expectation. The fluid balance of your body is the expectation that your body has. So the allostasis, or being in that allostasis, you could think of that as being in balance. So you're within the expected range of things. A stressor is any stimuli that goes beyond that preset range. So does it increase the speed or the intensity of any body system? So that can be the nervous system, that can be the peripheral nervous system, that can be the endocrine system, that can be the organs. So what what is a stress is these stressors, right? So it's that increase beyond, so that increased intensity, increased speed of the reactions of your body. So if I start to run, right? So if I'm on a treadmill or something or outside and I start to run, then that's a little bit beyond. I'm putting a stress on some different body systems that's beyond that allostasis piece. So that stress might be useful, it might be harmful, right? And that's what we'll talk about here. But that's what a stressor is. It's anything that's going beyond kind of that expected range of where you expect your body and your brain, your central nervous system, peripheral nervous system, and organ system, so endocrine system, to be operating within. So these stress has evolved for us to have a fight, flight, or freeze response. So to have us stop doing something, that might keep us safe. To run away from something, that might keep us safe. Or to fight something, that might keep us safe. So with this, here are some examples of different body systems that are related to it. So if we look at the brain, more attention and vigilance, you need more cognitive energy and working memory on the potential threat, right? On whatever stressor that is, whether it is a dog running out into the street in front of your car, whether that is a dog jumping out of the woods to attack you, right? You need more attention, more vigilance there. Pupil dilation, so your eyes getting more ready. And part of that is from the nervous system. Part of that is, well, from the nervous system, also talking to the hormone system of your body through like pituitary gland, decreased saliva production, increased breathing, increased heart rate and blood pressure to get more nutrients around in case you do have to fight or run. Decreased digestion, decreased filtration of getting toxins out of the blood, decreased food movement. We also see blood sugar and fat concentrations go up, basically giving more nutrients to cells all over the body because they might need it right now. But then we're also getting less digestion. So over a long period of time, you're trying to put more of that nutrients into the bloodstream, but you're taking less out of the actual digestive system to then put into it if you have more of a chronic stress. If this is like a dog jumps out and then you run back inside, you can calm down, things go back to normal, right? But if this is a long-term stressor or a chronic stressor, then you might see bigger problems because of these body systems working less or higher than they're supposed to be in that allostasis state. So reproduction activities go down as well. So these are things like production of gametes, for example. And then contractions in your muscles. So you might see someone actually shaking. So sometimes when I'm in my cold little room here, I'll realize, oh, I'm shaking, I'm shivering. My body has a stress beyond what it's supposed to be, right? It's supposed to be at this certain temperature level and it's a little colder. So when that happens, I'll go and turn on my little space heater. So that's nice to have. But there are a lot of body systems across the body that are related both to the brain, to the endocrine system, and the peripheral nervous system to communicate what's going on. But that's what stress does. It's affecting all over, not just the brain, not just your subjective experience, but also the body together. So this is the HPA axis, the hypothalamus, anterior pituitary, and adrenal cortex. So the hypothalamus talking to the pituitary gland, getting more adrenaline. So any kind of stress that we see can tell the pituitary gland to release more hormone. So usually this is hormones like adrenaline, cortisol, and with that, this leads to those different systems of the body slowing down or speeding up. So that's what it does. So to do that, to fly away, to freeze, to fight, this hypothalamus talking to the pituitary gland, telling it to produce more of these hormones across the body is what leads to a lot of these responses. So with that being said, we see here the hypothalamus, we see the hippocampus, we see the pituitary gland. It can tell the kidney to make more adrenaline. So you perceive a stressor, you release the hormone that tells the pituitary gland to send messages. Then the adrenal gland releases more of those into the bloodstreams through the different organs. There's multiple different organs and glands that do different hormones. Some kind of come from like one or two, but that's the basic idea. And then you metabolize that, you suppress immune function, you have all these functions that we just talked about in our body, and then it gets back, you get that feedback to your brain of, okay, we're at the level of those hormones now. The brain is working a little bit differently, focusing your attention on whatever stressor it is. So this system evolved to keep us safe, right? So let's talk about some kind of specific types of stressors. So we've got things like acute physical stressors, we've got chronic physical stressors. So we really evolved over time to try and handle these. So most of this system seems to be pretty focused on acute physical stressors. So things like getting away from a lion attack. After you have an accident, getting away, getting to a safe space, regulating your body when you are exerting yourself physically, and then for helping to support yourself after an injury so you can then get care. So, and that's one of that's a cool anthropological connection. One of the key pieces of things that they look at for people in for civilization and like, well, when do we say like this is human civilization as we kind of see it now? We kind of determine that as when did we start actually caring for people's bones that they couldn't have, that wouldn't have healed on their own. So like a femur, you're not gonna be able to get food for yourself with a broken femur and let that heal over time. You're probably going to die, right? So having a society that can help support you with that, that shows that humans were working together, helping that person get back on their feet, literally. So we can see that evidence in skeletons and things like that. But we also see chronic physical stressors. So illness can be you can have chronic illness, you can have an illness that lasts just a couple of weeks. Also starvation, obesity. These put stresses on your body, having not as much nutrients, too many nutrients, too much weight, not enough for what you need your body to do. Altitude exposure. So being at a high altitude for a long period of time, your body is using more of those stress hormones. So it's not in that allostasis, right? It's doing too many of these pieces. The systems aren't working kind of how it's designed to over time. And that's why some of these chronic stressors can be really damaging. Any of these chronic stressors, because it's using those systems, overusing those systems, underusing some systems, and that whole process can lead to long-term damage. In addition to these stresses, we have psychological stresses. So these might be real or they might be perceived threats or worries, but things like conflict, frustration, financial or debt, right? So grief or loss, there might be stresses around your career, getting tasks done in enough time, mortgage payments, right? Again, these can be acute, these can be chronic, but they do use the same system. So even if these are psychological stressors versus physical stressors, it's still using these same stress systems neurologically and biologically. So that's our stress system. So any stress has to go through the same system. So we also have social stresses, whether it be social isolation, like what we do to rats sometimes, bullying, other external forces that lead to racism, inequality, poverty. All of these can combine. So with these, as a social species, we really care about social interaction, and it is actually a protective factor. So having strong social bonds can, one, really help us to like get direct support for needs that we have to be met, but they can also just help us calm down, help us chill out, help us relax with friends. Right? So having social bonds is a big protective factor for stress. But in addition, these stresses combine, right? So some of these social stresses plus psychological stresses plus physical stresses, they can all be happening at the same time. Right? So you might have a biological stress of an illness. That means that you're not able to get to as many hours of work, which then means your worry about your bills coming up is even higher, just as one example. So I know talking about stresses can be pretty stressful. So sorry about that. So, with that being said, there is a level of stress that's good for us, right? So, our body, when we're digesting something, well, it needs to have the digestive system digesting. It needs to be active. It needs to be doing that so we can get the nutrients to our body. We call this in relation to physical or psychological or social stresses that like the good range of stress, eustress, or I've heard it called eustress or eustress. So whichever one is correct, it looks like this. This is the word. So this is when you are in a state of focused attention, you can still emotionally regulate, you can calm down, you can charge yourself up, and you're able to think rationally, but you are a little more engaged, right? So when you're taking a test in my class, I would hope this is where you're in. I would hope you're focused. I hope you're engaged. I hope that you feel like you're competent in doing this. You have that self-efficacy. And I hope you're not in this distress where you're at too much stress, that it's too intense, you're burned out, you didn't sleep the night before, you can't remember that thing from class. I hope you're not in that distress piece. I want to challenge you in that eustress piece. An interesting thing here that we see, well, before that example, also we see when you're below the proper stress level, you might be bored, apathetic, confused, maybe not paying attention. You might have seen this if you're like driving a car or around the same way that you always go. You might just kind of tune out so you're not stressed enough for that situation. Right? So we want people for learning, for engaging with others, for doing things to be kind of in this eustress piece. So how much stress is too much or too little varies on the person and varies on the task. So looking at this model, so just put in your notes, where along here would you think people set um, world records at? So one thing that we find is that when they're in practice, oftentimes in practice, so let's say you're an Olympic athlete, you're practicing today, your world championship for the Olympics is tomorrow. Usually your practicing is right about here. You're kind of at a balance of your focus and you're challenging yourself and you're a little bit stressed. And then interestingly, usually it's somewhere right around here where for you would generally expect it to be too much stress is where we're seeing world records being met. And so that goes to say maybe this person, because of their talent and their capacity, maybe their eustress is kind of expanding to it's capable of. It might also be a protective factor of being engaged and more having a good time with the audience and cheering. But oftentimes we don't see those world records being set. I mean, one, we don't count the world records if they're set in practice, we count them in the meets, but we see people perform a little bit better when they're under that additional stress. So, an interesting thing to think about. But again, this point where it's too much stress, where you're going too far beyond what your body, your cognition, so your central nervous system, peripheral nervous system, and endocrine system is able to do, that's going to be harmful. And the further up this too much side you're at, the worse it's going to be. Right. So shifting from stress, this is very related to another topic called flow. So this is a state of engagement. It's an enjoyable experience. It was one of the big first focuses of positive psychology because the flow experience is the feeling of being really engaged with something and feeling like time goes really fast, but you're really engrossed in it. So to you, both time might be going fast and slow, that you want to be doing it, you're having fun doing it, and you feel challenged It's a good feeling. So it's been called the optimal experience. I think there's other positive psych experiences that are really good too, but it's kind of been denoted as the optimal experience. And hopefully you've felt it at some time in your life. Maybe when you were doing art, maybe you were in a track meet, right? Maybe when you were trying to do something you really enjoyed doing and challenging yourself, maybe with other people around, things like that. One big key piece here is that if you're that you one big piece is you're trying to match your skill and your challenge level. So if the skill and challenge level meet and match, you might be in a state of flow. So I'm going to use the example of driving a car. Think back to when you were first learning to drive a car. So the very first time you actually sat behind a wheel and were trying to use the gas and brake. Maybe you had a clutch, maybe not, but your very first time trying to drive a car. Your skill level was probably pretty low, but the challenge of driving a car was probably very high. So you might have felt very anxious. Over time, maybe this was more worry. Your skill is getting a little better. The challenge isn't so big. And then maybe a month or two after you learn to drive, or maybe a year or two after you learn to drive, you could actually get some enjoyment from driving. You take a new road or a curvy road and you're really enjoying it. You're in the moment. You might have gotten a state of flow there. But now your skill for driving is probably higher than just like day-to-day driving on roads. So maybe you're bored or relaxed or even apathetic because you don't have much of a challenge to that skill of driving. And you can think of this with other things, whether it's playing an instrument, right? When you're starting it, you maybe try a song that's too hard. So you're alert and focused, or maybe you're anxious, maybe you're even worried because your skill is not matching your challenge. Versus maybe you get better and there's something that you're learning to play and it's just firing on all cylinders kind of thing. You might be in a state of flow. State of flows can last a few minutes, they can last hours, like um, probably the longest that we've measured is probably about 8 hours of a state of flow. And with that, your body is in this kind of heightened stress piece. So one, I don't know that it would actually be very healthy overall to have a longer state of flow, but most people it's like half an hour to a couple hours, like 2 or 3 hours in a state of flow, which is good. You probably need to use the bathroom, you probably need to get a drink of water, and sometimes when you're in a state of flow, you ignore those things. So this is another piece of the puzzle of human experience. It's very related to stress, but here it's also talking about what is your challenge, what is your skill level, and there are other things too. If you want some more detail here, I've linked a video in the notes of this slide. So go to it, download it, click on that link, but we won't actually watch that in class. But yeah, that is a flow state, very related to stress, eustress, and optimal performance. All right, so as we talk about this, your book goes into a lot more detail than we will cover here in this lecture. Basically, I hope you get a lot more of that detail in your reading versus the lecture. One thing that I did want to touch on with stress is the validity of these animal models. So I mentioned that a lot of these studies of animals were trying to look at very specific brain areas and how they're responding to emotion. Most of the studies that we're doing are things like fear and stress for animals because that's a little more immediate, a little easier to induce. But how valid are those? How valid are those measures of stressing out an animal in relation to humans? And so validity is basically the idea of are you getting a true or accurate measure? So if we see if what we call stress in a rat, is that actually stress in a human? That's what validity is. Is it actually the thing? Are we getting an accurate measure? Can we translate that to humans? Is it a valid measure? Means is it true? Is it accurate? So face validity, probably the worst type of validity, but one of the easiest to do. So it's kind of a first step a lot of people do in doing the validity of anything, whether it be a survey, whether it be a method of measuring rats and comparing them to humans. Face validity is, does it on its face, does it look like it would be the right thing? So you see, not the greatest scientific method. So do the behaviors that we see the rats doing, is their response, is their behavior, does that seem to be similar to humans? Does it look like it might be valid? That's face validity. It has its place. It's a good place to start. It's not an end place you should put for any kind of validity. It's a one additional piece of evidence. Construct and predictive validity, lots better. So construct validity, but if you don't have face validity, you might not even want to try and test construct validity, right? So it is a good step. So construct validity is, what is this construct? Are we actually measuring the correct construct? Are we measuring stress? Are we measuring fear? Are we measuring love in that rat? Right? Is what we think is happening in the underlying mechanisms neurally and in the body system, is it actually similar to humans? Is this rat's response more like a stress response in humans, or is it more of a fear response in humans? So what is that construct that we're getting at? The construct of fear, the construct of stress, the construct of love, right? Is it actually the correct underlying mechanisms? Some way that we test that is we put them through a situation that might be similar to a human. We check their brain areas, check with a human. Are those same brain areas active with the similar kinds of stress? To say, okay, yep, this looks like it's the same thing. This is stress. This is fear. This one we're not sure, so we're not going to use that method to test stress or fear. That's construct validity. Then predictive validity is kind of in its name. Can you predict using this? So we can stress out the rat. We can calm down the rat. Can we predict that this will also work in humans? That this kind of thing would stress out a human, or that this kind of thing would treat the human stress. So predictive validity, that's what that is. How good are we at accurately predicting in the human? And to do that, you actually have to try that with the human. You have to give the rat a drug, see if that decreases stress. Give the human the drug, see if that decreases stress. They both do. You've got some predictive validity. You've shown that the same treatment can work in both. So that's a really critical piece, is in any study, you want to make sure you're getting a valid measurement, so a true or accurate measurement. And that's a challenge with animals. And one of the main reasons we don't see a lot of animal research in like love and attachment, there is some, there definitely is, and there's different methods to do that. But it's a lot easier to get a more valid measure of things like fear and stress in an animal. So with all this being said and everything that you have read, hopefully you see how stress is related to so many body systems. So my face might be covering some of these. I don't know if I move my face, if it actually moves on your screen. But we see behind my face, there might be predictability, controllability, appraisal. These are the external and internal factors. But we have the limbic system. We've got a lot of midbrain areas. We've got things like the HPA. We've got the pituitary gland, hypothalamus, hippocampus, thalamus, hypothalamus, PGA, a lot of different areas. But in addition to that, we also have our endocrine system. So we have the organs that are secreting hormones, the organs reading those hormones and activating differently, and all of these in addition to the central nervous system, peripheral nervous system, and the endocrine system. That's the word I am looking for. All of these systems are interacting with each other, but they are also interacting with our DNA. So let's watch a video on epigenetics and stress. We've talked about this in week like 1, 2, or 3. So hopefully this is a little bit of a reminder. It's also a little bit more depth from those original videos. So hopefully you can remember that. And then we'll watch one shorter example of human stress actually affecting the immune system response.
You know something I love about biology? The depth, the complexity that we don't always recognize at first. I remember when I first learned about Punnett squares, I was in 9th grade and there were practice problems with eye color as the trait. But then, as we would explore more depth in biology, we learned eye color is actually very complex. It's a polygenic trait with many genes, and that trait is so complex that it really doesn't work like a simple Punnett square. In fact, even traits that we often treat like a one-gene trait, like attached or free earlobes, even that trait isn't quite that simple. So we even mention that in our pedigree video when we use it as an example. There are so many exceptions and details in biology, and they are fascinating to discover. They are still there waiting for you to explore them, even when you think you've mastered general biology. For a perfect example, something I not too long ago learned in a graduate level biology course, the topic of epigenetics. And I have to share it with you now, just like I did when I called my sister at 11:02 p.m. on a Wednesday night when I was covering it as part of my course. It's that interesting. Epigenetics. Consider identical twins. They have the same DNA code, right? But if one of them develops a disease that has a major genetic component, it does not necessarily mean the other one will also develop it, even though the identical twins have the same genetic code. Why? Recall your DNA has bases, and those bases code for physical traits like your eye color, your hair color, your height, and more. We learn about this in biology. But there's something else at work too. Epigenetic factors. I don't mean to sound so dramatic with my voice when I say epigenetic, but I really wish this had been something that I had learned about even when I was in high school. Although, to be fair, scientists are still learning a lot about epigenetic factors, and this field is already more advanced than it was when I was in high school. The root epi can mean above, which is fitting for the word epigenetic as above genome. You can visualize this word as factors that work on the genome and change how the genome is expressed. Let's talk about epigenetic marks. These can influence gene activity. One example is methylation. Methyl groups that are attached to DNA bases in certain locations, and they keep transcription from occurring. You remember transcription, right? It's part of protein synthesis. You have the DNA and you transcribe it to mRNA. That's transcription. You have to do that because the other part of protein synthesis is translation, and that's when the mRNA is used as a template, so the correct amino acids will be brought in to eventually make a protein. But DNA methylation can prevent transcription, so the DNA that is methylated will not be actively transcribed. Demethylation can remove methyl groups where those areas of DNA then could be actively transcribed. Epigenetic marks don't have to be on the DNA directly. They can also impact the histones. Histones are proteins that DNA will be rolled up and wound around, often compared to a spool for the DNA. Histones, too, can have methyl groups attached, which would be histone methylation, and this generally tends to prevent transcription. And we also want to mention histone acetylation, which is where acetyl groups are attached to certain parts of the histones. Now, when acetyl groups are attached, this causes a change to how tightly histones grip the DNA. The packing is more loose, and it allows transcription factors to access the DNA. There is also histone deacetylation, which removes acetyl groups, and this will cause the DNA to be more tightly wrapped around the histones. This intense packing will limit the access of transcription factors and enzymes involved in transcription, and this can be used along with methylation. Epigenetic marks like the ones mentioned can impact which genes are expressed, and that can make very big differences. Like what kind of difference? There are several examples in rodents that we have referenced in our video details. One example is how parental care that female rats have on their offspring, specifically whether female rats lick and groom their young or not, can affect the control of the expression of the GR gene. The GR gene codes for receptors that are involved in regulating the stress response in rats. Epigenetic marks, which in this case are influenced by the mother rat's behavior, can influence the transcription of the GR gene in the offspring. Due to whether this GR gene is frequently expressed or not, this can affect the baby rat's stress response and behavior even when they reach adulthood and have their own offspring. Another example is the agouti trait in mice. Consider two mice that have the same DNA. Only they don't look alike because one of them is a brown mouse with an average mouse size and the other is a yellow mouse with obesity. In the yellow mouse, a gene called the agouti gene is being actively transcribed. The gene is not being actively transcribed in its twin. It is actually methylated. And it turns out the active agouti gene can affect more than the color, as it also makes this mouse prone to being obese and potentially prone to specific diseases. Scientists found that the environment that a pregnant mouse is in and the diet that the pregnant mouse has can impact whether the offspring will have this agouti gene active or not. And if a pregnant mouse is fed foods that are high in methyl groups, Well, it turns out this can decrease the expression of the agouti gene in her offspring, even if the pregnant mouse herself fully expresses the agouti gene. Before I move on, 2 quick points I want to mention. First point is about inheritance. So when an organism's body cells divide in mitosis, the new daughter cells can inherit the same epigenetic marks as the original cells they came from. This can be inherited from cell to cell. Now, when a sperm cell and an egg cell combine to form a fertilized egg, which occurs in humans and many other organisms, epigenetic marks are often cleared. Except some epigenetic marks are not cleared and can therefore be inherited from parent to offspring in this way. See the further reading details. Number 2, I don't want to create a misconception that epigenetic marks are only something influenced by the external environment. So, for example, you have many different types of specialized cells. After all, most of your human body cells contain your entire DNA code, but generally your cells are actively transcribing certain genes based on the cell type, meaning your eye cells and stomach cells aren't actively transcribing the exact same genes. Many of these genes are actively transcribed or not actively transcribed due to epigenetic marks from internal cues, in addition to external ones. It's critical for human development, too. One last thing. You may wonder, why is the field of epigenetics so important? Well, as we mentioned earlier, many diseases that have a genetic component can involve epigenetics. Cancer can be an example, and cancer is one that personally has affected our family and many other families. We talk about cancer in our cell cycle video. And yes, with cancer, there is an uncontrolled growth of a person's own cells. And also, as we mentioned in that video, those cells are not functioning like they should. And by not functioning like they should, it can mean that there are genes that are actively transcribed that should be turned off, or genes that have been inactivated that should be actively transcribed. In cancer cells, the epigenetic marks are often not arranged as they should be. In a fascinating field known as epigenetic therapy, researchers are looking to advance cancer treatment options and treatment for a lot of other medical conditions too, by targeting those epigenetic marks acting on the genome. Overall, increasing our understanding of the epigenome, and by that I mean the genome and all of its epigenetic marks, will help us all better understand how our cells and bodies function. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
So pretty great video that does go into a little more depth than we talked about in chapters like 1, 2, and 3, but hopefully you remember some of those connections with epigenetics that we talked about before. So hopefully histone patterns and methyl groups are familiar to you still. So again, histone patterns is that winding and unwinding of the DNA, making it easier to access more of the DNA to transcribe or to do something with in the body. Right? And the methyl groups are turning on and off genes. So that is epigenetics, and stress is one of the main things that we've investigated. So stress is the main thing that we've investigated epigenetically in humans as well as in other animals. So other things have been looked at epigenetically too. So like protective factors like maternal warmth. But stress is probably the one at the moment currently that we know the most about. And most of what we see with stress is that it does bad things epigenetically. The outcomes of having stress maternally, like when you're in the womb, the epigenetic stress of your parents, of your parents' parents, has impacts on your genetics. And one example of that is decreased effectiveness of the immune system. So we'll watch this video. I might even speed it up. Again, if any of these videos don't play because I've had to delete them because of copyright strikes or things like that, please just click on the link and watch it. So especially that one on the video reminder of epigenetics, definitely give those creators a watch on your own rather than in this. But yeah, let's watch this one to give one other example of epigenetics affecting humans.
Turns out if your mom was really stressed, congratulations, now you're stressed too. Thanks, oh mama. Hey there, friends. Thanks for watching D-News. I'm Trace. Back in 1998, an ice storm hit North America. And if you live anywhere near the Canadian border, you probably remember it. I do. It was crazy. Everything was covered in ice from Michigan to Maine and Ontario to Nova Scotia. The weight of all that ice caused massive damage to infrastructure in both countries, and power outages dragged on for weeks afterward. Not to be caught out in the rain, so to speak, researchers at McGill in Canada found pregnant mothers who were without power and followed them and their offspring for 13 years to see how it affected them. Turns out, the North American ice storm of 1998 affected the kids' DNA. It used to be thought that if I was physically stressed in my body, it wouldn't affect my kids at all. If I was in a famine, that my kids wouldn't be affected by that. And then it turns out that's not true at all. In the 1990s, the field of epigenetics came onto the scene, and we discovered what happens to our parents will affect us, and what happens to us affects our kids, and so on down the line forever. This study, published in PLOS ONE, found that the children of those mothers had, quote, distinctive patterns in their DNA methylation. DNA methylation is the on-off switch for how the DNA gives its instructions. Whether to produce a bone or a red blood cell, for example, would have a different methylation. Alterations of that process change how the same DNA will be read by your body. In this case, a correlation appeared to exist between the length of time without power and subjective suffering of these mothers and the methylation of their children's DNA. 36 of the children tested showed the same pattern of methylation on their T cells, and that was the most stressed mothers. These cells are part of the immune system, so chances are the kids are going to have some kind of a different immune system response than their parents do. If you're wondering what that will mean, you're right there with the other scientists. Nobody knows yet. It's not like they can compare kids pre and post DNA methylation. They were born that way. But this is the first time ever in humans that science has...
At the time of that video's recording, it might have been the only piece of evidence, but I'm pretty sure there were other papers and they just hadn't done a wide enough lit review at that point. But it was a good, clear picture of one way stress can affect our epigenetics, and in this case, the immune system, leading to different effects. And this is, again, mostly what we've been looking at in relation to stress, and epigenetics are the kind of bad outcomes that come out of it. That being said, with stress, with trying to decrease stress, one of the big areas in therapeutic intervention, whether it be from a medical doctor, a physician, a nurse, a therapist, a psychiatrist, a social worker, oftentimes one of the big pieces of the whole therapeutic process is to decrease stress. So there's a lot of techniques and tools to do that. If you're in class, we will do a guided meditation in class. I find that can be especially helpful when we're talking about stress. It often reminds you and triggers the stresses that you have in your life. But it's also just a good thing to do. So meditation is one example. There's a lot of different ways you can decrease stress in your life. So active problem solving being the best thing to do, but not everything can be solved by active problem solving as well. So that link is here in the slides. So feel free to follow along with that guided meditation. If you've done meditation before, you don't need to do this guided meditation. Do some other stress and relaxation techniques that you know. So, but if you're in class, we will do that together as long as we have time. But yeah, a guided meditation is a really useful first step if you've never done any meditation, because it kind of gives you instructions of exactly what to do. All right, and this specific version, maybe I'll pull it up on the screen. This specific version of a guided meditation is actually something that I did with colleagues in graduate school to see if this 7.5-minute video, would this help decrease stress, decrease anxiety, increase things like prosocial behavior, and how long would those effects last? This 7.5-minute video doesn't even really last 15 minutes for continuous decreased stress for most people, but it starts to teach you how to do it, and it does have an immediate effect on your stress, which is great. But that's also one reason why a lot of meditation programs and people that encourage meditation encourage you to do it often, because you can really help with that plasticity in your brain as a response to stress. Just like you have triggers that induce stress that we've been talking about, all sorts of things. So we talked about psychological stresses. We talked about psychological stresses, we talked about social stresses, we talked about physical stresses. Just like you have those stresses those stresses might trigger a specific stress response, you can build into your neural networks responses to that stress to calm yourself down. So whether it's take a break, whether it's take a step back, whether it's to bring in the cognitive abilities, whether it's to then, oh, I can tell I'm at a certain stress level, let's go do some meditation, let's go do some art, whatever it might be, you can build that into your neural networks because of plasticity. All right, now we'll move on from stress to trauma specifically. So trauma is a specific kind of response. So they are not the same. There's a lot of overlap here, but stress and trauma are distinct definitionally. And so researchers have very specific definitions of stress and very specific definitions of trauma. You might hear just some random layperson like talking colloquially about stress and trauma and use them interchangeably, but a researcher is going to use them differently. Now, why I say a researcher might use them differently, a therapist might use them interchangeably in some cases and for some reasons. Also, depending on if you're, say, submitting a grant and what you're really working on is stress because it has a link to trauma, you might say stress can lead to trauma, and so we want to reduce trauma because that's kind of a bigger, intense thing that maybe a funder would be like, oh yeah, I do want to help decrease that kind of trauma. So stress, again, goes back to what we discussed in a little bit earlier. A stressor is something that increases the speed or intensity of your central nervous system, peripheral nervous system, endocrine system beyond the kind of allostasis that it's supposed to be in, right? So a stressor gets at the increased speed, increased intensity of those systems beyond the standard range. A trauma is specifically an emotional response to a traumatic stressor. And so for it to be considered a traumatic stressor, it has to fit a couple criteria. It has to be something that seems to be more intense in potential threat to you. So being involved in an accident, being a victim of crime, in a natural disaster, physical or emotional abuse, severe neglect. And so usually there's like a qualification system here, like severe or extreme, or experiencing or witnessing violence, being part of war, being part of a genocide. These would fit the definition of trauma. So these are basically specific types of stressors that get to the level of what we define as trauma. So there's a definition linked there too. I also have an article linked in the notes of this slide. So if you want to look at that as well, that can give you a better sense of what do we mean by stress versus trauma. So long-term stress, or some of these stressors, can then be a trauma. So that specific emotional response, a specific response in your body, we define as that trauma. So some stressors might be very stressful, but might not be a trauma. So there's lots of stressors. Me running on the treadmill is going to be stressing my body, but that's not going to be a trauma. Getting hit by a car while I'm running down the road, that's going to be way more likely to be a trauma. So hopefully that is a little more clear now as we continue on. But again, the researchers are going to have very specific definitions. A layperson might not, it might not be important to really distinguish that with them. And a therapist might even use them interchangeably depending on the audience that they're talking to, right? So talking to a person in a chair in front of them, they might use them interchangeably. It might be useful in that situation. If they're at a research conference, they probably wouldn't be using them interchangeably. They'd be a little more specific to that. All right, so let's talk about specifically the autonomic nervous system. Most of what we've talked about today in the class has gone into the central nervous system and the endocrine system, but let's talk more about the autonomic nervous system and how it is related to trauma. Again, if any of these videos don't work sometime, please click on the link on your own. I put a lot of videos in the lecture today, so if any doesn't work, click on it yourself, because sometimes I have to delete them because of copyright strike.
Over half of us have a chronic disorder such as high blood pressure or autoimmune disease. Rates of anxiety, depression, PTSD, and addiction are skyrocketing. Why? The roots of these issues and more can often be traced to trauma, adverse childhood experiences, chronic stress, and ultimately, nervous system dysregulation. Meet your autonomic nervous system. Let's call it ANS. ANS takes care of a lot of your automatic functions, like your heartbeat, digestion, and body temperature. ANS also manages your survival and stress response, working to keep you alive when your life is in danger. ANS functions as our built-in detection system, constantly scanning our environment for cues of safety and cues of danger. As ANS scans the environment, it has 3 general responses or states. Safe. You feel calm, relaxed, and connected to those around you. Mobilized. When ANS detects danger, it sends a command and your heart rate and breathing increase, adrenaline and cortisol are released, and blood rushes to your muscles so you can handle the threat. This is our fight-flight response. Immobilized. When ANS detects that the danger is so great that you can't fight or run, it shuts you down. In this state, our heart rate, blood pressure, and body temperature decrease, and pain-numbing endorphins are released. ANS does all of this automatically without us thinking about it. ANS doesn't just use these states for survival. It uses them to navigate through the world each day. When ANS functions well, it moves fluidly from one state to another, 1 minute mobilized and ready for action, and the next resting and recovering. ANS will often blend states together. When we play, ANS combines the mobilized and safe states, and when we are intimate with loved ones, it combines immobilized and safe states. When ANS can stay flexible and fluid like this, it helps us manage and become resilient to stress and negative events. We're able to bounce back and move on. Unfortunately, when we experience trauma and chronic stress, it can keep ANS from functioning in a healthy, regulated, and resilient way, and can keep us stuck in states of survival. A friendly get-together can become frightening. A simple meeting at work can become threatening. For those with a history of trauma and chronic stress, the ANS detection system often becomes faulty, constantly signaling danger even when we are safe. It's like ANS is an alarm system, constantly signaling fire even when there's no smoke and no flames. Consistently living in these survival states can be debilitating, and we often develop adaptive strategies like using drugs, alcohol, food, work, or sex in an attempt to bring regulation and temporary relief. Understanding how trauma impacts us is critically important. There is a whole spectrum of experiences that can be traumatizing and adversely impact ANS, like accidents, assaults, and natural disasters, which are often called shock traumas. There is also developmental or relational trauma, when we experience chronic adversity, abuse, neglect, and lack of safety while growing up. Many other experiences can be traumatizing, including chronic stress, medical procedures, and adverse community environments, like poverty, discrimination, and violence. Additionally, new research in epigenetics shows us that trauma can get passed down genetically at least 3 generations. In the past, we've thought about trauma as events that happen to us. We now know that trauma is an experience, not an event. It's what happens inside of us as a result of what happens to us. It's our response to the event rather than the event itself. Over 20 years ago, Kaiser and the CDC launched a groundbreaking study of over 17,000 patients that showed a direct link between adverse childhood experiences, or ACEs, and long-term health and wellness. In the study, 2/3 of participants reported at least one ACE. Over 20% reported 3 or more. When participants reported 4 or more ACEs, this corresponded to an increased chance for heart disease, cancer, drug abuse, and more. With 6 or more ACEs, life expectancy decreases by almost 20 years. We are learning that many physical and emotional symptoms may emerge from a chronically dysregulated ANS. When ANS gets stuck in survival states, our biology shifts its focus from the tasks that keep us healthy, happy, and thriving to surviving the immediate perceived threat. Many conditions and symptoms that are chronic and difficult to diagnose and treat can be attributed to a dysfunctional ANS. Our childhood experiences can also keep us from connecting with others. This is vitally important because as children, our number one survival priority is to attach to caregivers. When the people responsible for our safety aren't safe, and we are living in chronic states of unsafety, ANS doesn't get wired right. The part of ANS that judges what is safe and what is not becomes faulty. If intimacy and connection were unsafe as a child, as adults, we'll often unconsciously reject attempts from friends and partners to connect. Even though intimacy and connection is what we want, ANS feels it's unsafe and won't allow it. Trauma compromises our ability to engage with others, replacing the need for connection with the need for protection. When there has been trauma, ANS can no longer differentiate between our unsafe past and our now safe present. ANS can't turn off the need to protect, even though we are now safe. So what can we do when ANS becomes dysregulated? How do we recover from trauma and develop a healthy, regulated, resilient nervous system? Fortunately, we can retrain ANS to feel safe again. This is best done with the help of others. Each one of us has an ANS, and our ANS is constantly communicating with and attuning to the states of others. We autonomically mirror the states of those around us. This is called co-regulating. We see it in herd behavior. If one animal senses danger, the entire group becomes more alert, increasing their chances of survival. We're exactly the same. When we're with others who are stressed, angry, or depressed, it makes us feel worse. When we're with others who are calm and happy, it makes us feel better. Connecting with others who are safe, attuned, and present is the best way to restore a healthy ANS. For those struggling to recover from the impacts of trauma, there is an emerging field of innovative clinical therapies that have been developed to reestablish safety and regulation to ANS. We're also learning that many of the activities we intuitively know make us feel better, like spending time in nature, practicing yoga, dancing, helping others, and more, can help ANS become more regulated and resilient. Healing from trauma and finding release from being stuck living in survival states comes as ANS becomes regulated, increases its capacity for resilience, and regains its flexibility. It's not about being calm all the time or mobilized all the time. It's about having a flexible and resilient nervous system that can accurately assess safety and danger and responds appropriately. We're truly resilient when we can fluidly move from one state to another. For those living with the impacts of trauma and chronic stress, becoming unstuck is like beginning a new life. For the rest of us, understanding how our nervous system states guide our behavior can help us become happier, healthier, and more empathetic human beings. Collectively, we have an epidemic of social issues that are rooted in trauma. If we can do the work to heal past traumas and build healthy, regulated nervous systems as individuals, families, and communities, we can end the cycles that continue to reinforce our greatest challenges and create a safer, vibrant, and more connected world.
Lots of detail in that video about trauma specifically. And one thing that I really like about the end of it is that it gets into the treatments, which is where I want to get into for the end of our lecture, as well as going into your hybrid activity for the week. So these aversive childhood experiences, this is a piece of the aversive childhood experiences questionnaire. You may or may not have ever seen that. Feel free to pause really quickly. Because basically, however many of these, this is an assessment that tries to get at what stressors did you have, how likely might it be that you will have trauma right now, and then based on these aversive childhood experiences, like it was saying, the more of these that a person checks off, the higher the risk factor. So that is one thing that in developmental psychology especially, we look at those risk factors, those protective factors. We try to emphasize protective factors, encourage them, decrease risks, so that it can help people thrive and develop optimally and in a thriving way. So that's what these aversive childhood experiences and these kind of looking at stressors and is it likely that you have trauma is about. So they alluded to this in the study. I think this is a really good graphic right here about aversive childhood experiences. Again, pause for more detail on this. But with this, the more of these aversive childhood experiences, the more of these stressors, the more likelihood of trauma that you have, the higher the likelihood of suicide attempts, the higher likelihood of drug abuse, the higher likelihood of diseases and immune system compromisation, these kinds of things. So these are the negative potentials that can affect things like epigenetics as well. With that being said, this is a really important thing to consider when treating people is what kind of stressors have they had? What is their experience? What is the potential for trauma that they might have? And so an ACE-informed approach is really a trauma-focused approach, right? There's a lot of words or a lot of terms for this. The idea is to be aware of that, help people work through some of those stresses and traumas, help give them more protective factors so you can have more safe, stable, nurturing relationships. You can build relationships between parents and children. You can get more supports in a community. So you can give concrete supports like money, for example, or a safe place. You can give support in other ways that's not as direct. Right, so this kind of trauma-informed care is more holistic, looks at things overall because there's a lot of protective factors. And this is one big thing is how many, if we add more protective factors, then it will decrease the negative impact of those risk factors, of those aversive childhood experiences. So something like maternal support, so helping train a mother to be more responsive to the needs of the child, that might be helping them understand how is that child communicating with their crying or their acting out or whatnot. So helping that parent to then be more responsive, or to an adolescent to be more responsive, help and work together so you are working together kind of thing. That can help support a person through these things. So with that being said, emotion, stress, trauma, we've talked about a lot today, and I want to lead into some of the kind of specific treatments. So for trauma, especially, these are a lot of things that are used for treatments of trauma. So one that you'll focus on in your hybrid activity is art therapy, but being with other people, trying to build your own skills, even things like EMDR, and psychodrama, having rituals and community, a lot of these things can really help to, one, regulate your autonomic nervous system, create new neural pathways that you can use when facing a stressor, when a trigger for a trauma might come up. And essentially that is the idea about all these treatments, is to use the systems that we know and build in new systems for your body to go around, right? To build new systems that can have a maybe more adaptive result in the end. So here is specifically a lot of detail on art therapy, and you'll get into a little more on art therapy in your hybrid activity. If you're in a hybrid class, versus we'll do this in class if you're not in a hybrid class, some activities related to art therapy. Yeah, the idea here is that you are using different areas of your brain as you're thinking about a trauma or even potentially being triggered by a trauma. You're engaging more neural networks. So you're engaging not just those couple core base emotion networks for fear or for grief or for attachment or for desire. You are bringing in other neural networks. So your brain is learning how to process things a little bit differently. And that's a big thing about the plasticity here in the brain for whether it be stress, whether it be fear, whether it be rage and anger, is to build those new connections in the brain. So that is it for these chapters. I know there's a lot more reading that we could have done, and there's a lot more things in your reading, especially in that chapter 12 on stress that we didn't cover in here, but hopefully you did go over those in your chapters because it is all useful to fill out your picture of what humans are experiencing. So double check your syllabus for the next couple chapters we will be reading so you are in line with the class.